around once more. We start maybe, you signed, right? Yeah, we start maybe here. And then you can circulate around so that everybody has a chance to sign. Well, yes, as said, let's start with the science. The first speaker, first lecturer, I should say, is Chuck Farah from around here. We're not going to introduce the lecturers in detail because they can do that themselves if they like. Just want to hear the science. So, stage is yours.
Thank you, Ralph. Thank you, Fernando, for inviting me to give these introductory talks. So, unlike you, most of the people in this audience, I'm not a physicist. I'm a biologist / biochemist. And I was told that while a lot of you, seeing all the posters here, there seems to be a lot of biochemists in the audience, but I was also told that there are many people from just a pure physics background, and that they would like to have a couple of introductory lectures on macromolecule structure and how biochemists look at the problem of protein structure and protein function. So that's going to be a topic basically of my 3 talks today. Okay, so the first talk is going to be on just protein structure basics. Okay, probably everyone has heard about this central dogma that says that the genetic material codes for RNA, and RNA molecules code for proteins, and proteins are polymers of amino acids, and they fold up into specific 3-dimensional shapes that gives them specific functions, often catalyzing enzyme reactions or binding to other molecules. And this 3-dimensional structure is what dictates the protein's function in the cell. So while we do have a genetic code that goes from the DNA sequence to the protein sequence, we don't have a code that goes from the protein sequence to the 3-dimensional structure, and we definitely don't have a code that goes from the 3-dimensional structure to its specific function. Okay, so these protein folding, this relationship between protein folding and structure and function are still some of the most interesting and deep questions in biochemistry and biology. So this is basically our vision, biochemist's vision of a cell. This is a bacterial cell, and you can see that the inside of it is just jam-packed with macromolecules. So when we often model these systems in aqueous solution and dilute aqueous solution, we have to sort of keep in mind that maybe in the cell the conditions are much different. Okay, we've got probably a very viscous solution with large polymers, in which we have lots of interactions, and the diffusion in this kind of condition, in this kind of environment, is much different from that in aqueous solution in a test tube, for example. Okay, and this is just like a zoom here. So we have protein molecules, we have water molecules interacting with these large polymers of nucleic acids. Okay, and we can represent molecules in different ways. There's some common coloring scheme conventions. So normally we have carbon atoms shown in white or green or black. Hydrogen atoms, if they're shown, will be usually white. Nitrogen blue, oxygen red, and sulfur and phosphorus will be yellow or orange. Okay, so that's the kind of coloring scheme that normally biochemists will use. And this is just some depictions of some macromolecules, the double helix of DNA, messenger RNA, transfer RNA, a ribosome that's made up of proteins and RNA molecules folded up together. Okay, so proteins are polymers of amino acids, and there are 20 basic amino acids that are coded by the genetic code, which is universal in almost all forms of life. all of these amino acids have a common structure. Okay, they have a nitrogen, an amino group here in blue, a carboxyl group, or actually a carbon with an oxygen atom attached, and a central carbon atom, which is called the alpha carbon, okay, to which is attached the side chain. And the side chains can be different. They can be small side chains, like just a methyl group here. Here, the hydrogen atoms are not shown. So here we have a carbon, which is, for alanines, just a methyl group. And then we have some larger, bulkier side chains that have different characteristics. They can be acidic, basic, hydrophobic, hydrophilic, etc. Okay? And so when we talk about different levels of structure for these polymers, for these protein polymers, we often talk in primary structure, which is just the sequence, the covalent structure of the polymer, which is the sequence of the amino acids, okay? Then these local sequences can fold up into common structural motifs, okay, which is called secondary structure. This is one example of it, which is called an alpha helix, which is a right-handed helix, okay? And then these secondary structure elements in one chain will fold up in three-dimensional space in a specific topology, okay, which is called the tertiary structure. And in many proteins, the biologically relevant form is actually a complex of more than one chain that come together in a quaternary structure, which is basically how these three-dimensional, or how these individual chains interact with each other. Okay? So we talk about different levels of protein structure: primary, secondary, tertiary, and quaternary. Okay? So, how are these amino acids connected to each other chemically? They're connected through what we call peptide bonds. Okay? So, a peptide bond is basically an amide linkage, which is created by the condensation of a carboxyl group of one amino acid and an amino group of another amino acid with the elimination of water. Okay, so chemists will recognize this kind of reaction. In the cell, this reaction it's not a direct condensation reaction. It's a more complex activated reaction that occurs in the ribosome, but essentially the end product is this amide linkage here, which we call a peptide bond. Okay, so here we have one amino acid here that is attached by a peptide bond to the preceding amino acid, and by another peptide bond to the next amino acid, and that allows a chain of amino acids to make this linear sequence of amino acids, and then this will fold up to make a three-dimensional tertiary structure. Okay, that is the biologically relevant form of the molecule in the cell. Okay, so these higher levels of structure, the secondary structure and the tertiary structure and the quaternary structure, are determined by thousands, or depends on how you define them, thousands or tens of thousands of very, very weak non-covalent interactions between specific groups in the primary structure. So all of these atoms and these chemically functional groups here can interact with other groups in the molecule, okay? And the sum of all of these interactions, these non-covalent interactions, will determine the final tertiary structure, okay? And so what are these non-covalent interactions? There's several different types: hydrogen bonds, van der Waals interactions, dipole-dipole interactions, an effect that we call the hydrophobic effect, which is really more of an entropic effect, okay? There's an exception to this rule that these are non-covalent interactions. There are some covalent interactions, covalent bonds made between different protein chains which are disulfide bonds. So they're covalent bonds between sulfur atoms and cysteine side chains, okay, that can make a strong covalent linkage between one molecule and another molecule, okay? So this is just basically showing the strength of these non-covalent interactions, okay, in terms of bond energy in kilocalories per mole. So you can see that these hydrogen bonds and van der Waals interactions and ionic interactions are all less than 10 kilocalories per mole. But there's literally hundreds or thousands of these interactions occurring, okay? And the difference between their interactions, these intermolecular reactions, or intramolecular reactions, that is the difference between how the sum of these interactions between groups in the same molecule versus their interactions, their potential interactions with the solvent that surrounds them, will determine the stability of this three-dimensional formula. Okay. So if we look at it more closely at the three-dimensional structures of proteins whose structures have been determined experimentally, we see that in the interior of the molecules, they're very, very closely packed. Okay, so the interior of a protein is usually sort of like the interior of an oil droplet. Okay, so there's not very much space for solvent molecules to come in and out. Okay, this close packing increases the number of these van der Waals interactions. Okay. So these van der Waals interactions are maximized, and that's a very, very strong contribution to the final form. So this is the kind of close packing that we see when we look at the interior of a protein molecule. Okay. Now, what are the if we want to model the possible, if we want to consider from a theoretical point of view the possible conformations that a protein molecule can adopt, okay, there are some considerations we can take into account. Firstly, if we look at the peptide bond, okay, we can see that there's a lone pair of electrons here in the nitrogen, the amino group that aren't shown, and it can be delocalized. So we have this resonance structure between the electrons in the nitrogen in this single bond here and this double bond, this carbonyl group. Okay, so we have a delocalization of this pair of electrons to make a pi bond between the carbon and the nitrogen. So this peptide bond, even though it's normally written as a single bond, okay, in fact it has a significant double bonded character, and that restricts its rotational freedom. So in fact, these peptide bonds are always found in either the cis or the trans conformation. And for steric reasons, it's almost always found in a trans conformation. Okay? There are some specific cases where you can find cis peptide bonds. Okay? So that basically means that the carbon alpha atoms of consecutive amino acids and the peptide bonds, so these 6 atoms here, the carbon alpha, this carbonyl group, this amide group, and the next carbon alpha, they're all in the same plane, okay? Either in a cis or in a trans configuration. And that's usually a relatively good, valid approximation, okay? So this angle here is either 0 degrees or 180 degrees, plus or minus 10 or 15 degrees, okay? So that means that we can look at the covalent structure as sort of a linkage between a whole bunch of planes, okay, that are attached by these central alpha carbon of each amino acid. Okay, so the relevant conformational flexibility here, if we're just looking at the main chain, okay, we define this as the main chain here, and these are the side chains. We can look at the main chain as really a whole bunch of linkages that are defined by these angles here, these rotational angles at the carbon alpha. Okay, so these angles here are called phi and psi. Okay, phi and psi. And so the phi angle is between the nitrogen and the carbon alpha, and the psi angle is between the alpha carbon and the carbon of the carbonyl group. Okay, and we can define the conformation of this amino acid by this combination of these two rotation angles. Okay, and you can see that since these are single bonds, there should be free rotation about these two angles, but there are some combinations of phi and psi that will create steric clashes between groups further along the chain, further ahead and further behind the chain. Okay, so there are certain combinations of phi and psi that are not available. Okay, and these can be calculated theoretically because we know the van der Waals radii of all of these atoms, so we can calculate theoretically what combinations of phi and psi should be available and which ones should be disallowed. Okay, and this landscape of possible combinations of phi and psi is well defined by what we call a Ramacandra plot, okay, which is basically a two-dimensional plot of phi angles here, minus 80 to 180 degrees, and psi angles here, minus 180 to 180 degrees. And the allowed regions are shown in yellow and red, okay? And all the other regions are disallowed, basically, because they create steric clashes, okay? So this reduces the amount of flexibility theoretically available to the polypeptide chain. And you can see that there are basically three islands of relative stability, of conformationally allowed regions, okay? And these are, for historical regions, called the right-handed helix, or right-handed alpha helix region, the beta sheet region, and the left-handed alpha helix region. You can see this beta sheet region is it's sort of like in a more extended region where the angles are close to 180 degrees, both for phi and psi, and the right-handed alpha helix has a phi angle of near 180 degrees, but a psi angle around minus 60 degrees, okay? We also have this left-handed alpha helix region, but it's a much smaller region, and therefore it's not that well populated, okay? So if we actually look at a protein, okay, and have a dot for every single amino acid in that protein, and see where these phi and psi combinations are found, you can see that almost all of them are found in these theoretically allowed regions. Now, there are some that seem to be breaking the rule, but these are usually special amino acids, glycine amino acids. Glycine is an amino acid that only has a side chain as a hydrogen atom, and therefore the conformational space available for glycine residues is much greater than this standard Ramacandra plot. Okay? So, so we have basically these two islands here of allowed conformational space. And what does this result in? What's the consequence of this conformational restriction for the polypeptide chain? It basically says that there are two major ways in which the polypeptide chain can fold up locally, okay? And that's what we talk about when we talk about secondary structure. So one very common form of secondary structure is called the right-handed alpha helix, okay? Which is basically it's stabilized, and this form, as well as being in a conformationally allowed region of the Ramacandra plot, it's stabilized by these hydrogen bonds here between carbonyl groups, which is a hydrogen bond acceptor of an amino acid at the I position, and a nitrogen group, an amide group, NH, which is a hydrogen bond donor at the I plus 4 amino acid. So this is I plus 1, I plus 2, I plus 3, I plus 4. This is the nitrogen of this I plus 4 residue, and it is pointing back at the carbonyl group making this hydrogen bond. So this alpha helix is stabilized by these hydrogen bonds, okay? Now, these hydrogen bonds, you can say, well, why are these hydrogen bonds particularly more stable? Because hydrogen bonds theoretically could be made with water. So this protein chain could unfold, and all of these hydrogen bond acceptors and hydrogen bond donors could be satisfied by making hydrogen bonds with water. Okay, but as well as these hydrogen bonds, there are van der Waals interactions occurring between groups in this main chain that are stabilizing this conformation. And also, this structure can be buried in the interior of a protein where there is no access to water. Okay, so this can stabilize a three-dimensional structure in which a significant percentage of the amino acids are not interacting anymore with the solvent, but they remain able to make these hydrogen bonds, okay? And therefore, the difference in the energy between the folded structure and the unfolded structure is reduced, okay? So this is very important for the folding of proteins into a globular form, where there's a significant fraction of the amino acids are buried in the interior of the molecule, and they have to make intramolecular hydrogen bonds because the possibility to make intermolecular hydrogen bonds with water molecules in a solvent is not available. Okay? This is also true for beta sheets. So this region of the Ramacandra plot, so if we have a lot of consecutive amino acids that have phi and psi angles in this region, it will be a more extended form, okay, making these what we call beta strands, okay? And these beta strands, they can interact with other beta strands, either in an anti-parallel or in a parallel manner, okay? And these strands are stabilized, or these sheets. So if you have two or more strands interacting with each other, they will form what we call a beta sheet, okay? And these beta sheets are stabilized by hydrogen bonds between amide groups and carbonyl groups of the neighboring sheets, of the neighboring strands. And on these strands, the amino acids are alternating above the plane of the sheet and below the plane. So here we have a side chain above the plane, so it's coming out of the page here, out of the screen, and then the next amino acid, the side chain, is below the plane. Okay, so it's like a zigzag pattern. Okay. On the same the same point of view, we can look at an alpha helix, and you can see the side chains are all pointing outside of them, radially, and actually radially and a little down towards the amino terminal of the alpha helix, but they're basically pointing out radially from the axis of the alpha helix, and that makes this alpha helix able to accommodate almost any amino acid sequence. Now there are some amino acids that are particularly favorable to forming alpha helices, and there are some amino acids that are not very common, that are not commonly observed in alpha helices. But in general, this arrangement with the sequence pointing outside, the side chains pointing outside, allows almost any sequence to be accommodated in an alpha helix. And that's the same case for a beta sheet. The side chains are pointing above and below the plane of the sheet, and therefore, in general terms, okay, it can accommodate almost any sequence. Okay? So this is an anti-parallel sheet, so we have one strand going from the N terminus to the C terminus, and this going from the N terminus to the C terminus in this direction. But we can also have parallel beta sheets. Okay? For the same reason, these beta sheets can be buried in the interior of a protein in a hydrophobic environment. Okay? Because they satisfy the energetic requirements for these hydrogen bond donor and acceptor groups to form intramolecular hydrogen bonds, okay, in a hydrophobic environment. This is also important for proteins that go through membranes, okay, so membrane proteins that go through the hydrophobic environment of a membrane. They have to satisfy their hydrogen bonding requirements by intramolecular interactions, because there are no water molecules available in the membrane, in the hydrophobic environment of the membrane. Okay, so how do we represent these molecules in like a computer graphics program? So you're going to, you have biochemists to summarize or to simplify their representations of these complex molecules that have literally thousands of atoms. Okay. They will adopt several sort of visual conventions, okay? So like I said before, often the carbon atoms are shown in green, okay? The nitrogen atoms are almost always shown in blue. The oxygen atoms are almost always shown in red. The hydrogen atoms are often left out, okay? So they're, in this case here, we've got just two amino acids, an alanine and another alanine. Okay, these are the side chains of the alanines, and we don't have any hydrogen atoms. So we assume that the hydrogen atoms are there, but we're not representing them just to simplify the image. Okay, so if we make an alpha helix here with the side chains, okay, we can see that it's already getting a little complex. It's hard to follow the chain. Okay. And now we're sort of like rotating this alpha helix, and we're looking at it from an axial view. So now we're looking down the helix axis, and we're seeing the side chains decorating the perimeter of the alpha helix, the outside of the alpha helix. Okay. We can represent it with or without the side chains. So if we remove the side chains, now it's become simplified. Okay. And then we can also simplify it even more by just representing it as a ribbon diagram or a cartoon diagram, okay? So this is the same alpha helix shown, side view, and axial view, okay? So this is a very, very common way of representing protein polymers, the polymer chain. You follow the trajectory of the main chain, okay, by just following these ribbon or cartoon diagrams, okay? And they'll often have a arrowhead at the end, at the C-terminal end of the chain, so you know in which direction the chain is going. Okay? So just like we can represent helices in this ribbon diagrams, we can also represent beta strands or beta sheets. Okay? So this is basically like what we call a beta hairpin. It looks like a hairpin because it's got a beta strand and then a very short term that takes up 4 amino acids usually, and then we have another beta strand, and we have these hydrogen bonds that are stabilizing these two beta strands together. And you see that they're in anti-parallel orientation just because the arrowheads are pointing in opposite directions. Okay? So here we assume, what do we assume when we look at this? Okay? So a biochemist will look at this and he knows intuitively that this really means this. Okay? That this is the backbone, so this here was basically this here. This is just showing the beta strands of the backbone, okay? We can add the hydrogen atoms if we want, and now we can actually see the hydrogen bonds between the amino group, the amide group, and the carbonyl group of the neighboring side chain, okay? So they're alternating back and forth. You can see that this is stabilized by a whole bunch of hydrogen bonds between oxygen atoms and NH groups here. Okay? And here, this is the same beta hairpin with a side chain. Okay? And we can also represent this with space-filling models. Okay? So we can represent each atom with a sphere that is represented by its van der Waals radius. So it's like a hard sphere. We know that nothing, once another group penetrates this sphere, it will be overwhelmed by repulsive interactions. Okay, but we can also sometimes the closest approach is determined by the size of the other molecule. Often these other molecules are water molecules. Okay, so what we can do is we can represent this surface by what we call a solvent-exposed surface. Because water molecules have a radius of approximately 1.4 angstroms. So if we assume that they are like a sphere of 1.4 angstroms, how close can a sphere of 1.4 angstroms get to the surface of this molecule here? So that can be so we can say that, well, even though the van der Waals radii, there's a deep pocket here, a water molecule of 1.4 angstrom radius will not be able to penetrate it. So how deep can that water molecule go in there? And that's represented by this surface. Okay, so often there are different ways of representing the surfaces of these proteins. Okay, now so we have this primary structure, which is the sequence. We have these common secondary structures that are relatively stable and they're conformationally allowed because they reduce. On the one hand, they reduce steric repulsions between neighboring groups, and they're stabilized by hydrogen bonds. Okay. These secondary structures can come together to make what we call common structural motifs. Sometimes they're called super secondary structures. Okay, so when we have one or two or more secondary structures that come together in a specific topology that seems to be found in many different proteins. So there's sort of like a classification. So when we can sort of like give a name to this kind of arrangement, and the name immediately renders in the mind of the audience what you're talking about. Okay, so this is like this is what I call the beta hairpin here, that makes an antiparallel sheet, and this is what we call a beta loop, super secondary structure. So we have this beta strand here, then a short loop that comes around and allows another beta strand to interact with the first beta strand in a parallel manner. Okay, so this is often called just basically beta loop beta. Okay? Sometimes this loop can actually have an alpha helix here, so in that case it would be called a beta alpha beta loop. Okay? And these beta alpha beta units can interact with each other to make a continuous, like barrel-like structure. Okay? So sometimes the barrel can close upon itself to make what we call an alpha beta barrel. Okay? So these super secondary structures can sometimes interact with each other in a common motif form to make larger structures. Okay? There's another kind of motif called an alpha-alpha corner, so it's just an alpha helix with a loop and then another alpha helix. There's a version of this called an EF hand. EF hands are a specific version of this alpha-alpha corner in which the loop has specific amino acids, and it's like 12 amino acids, and at specific positions there are amino acids whose side chains will coordinate calcium or magnesium atoms, or ions. So these EF hand proteins are often associated with calcium or magnesium binding, and they perform very, very important functions in the cell. Okay? Sometimes alpha helices can come together in what we call a coiled coil. Coiled coils are stabilized by hydrophobic interactions between hydrophobic amino acids at positions A and D in what we call a heptad repeat. Okay, because if you look at the sequence repeats itself every 7 amino acids. So at positions A and D, if we have hydrophobic amino acids here, this surface of the alpha helix will be lined with hydrophobic amino acids, and it will preferentially interact with a hydrophobic region of another molecule. So often another alpha helix that also has this kind of repeat will interact with it. Okay? So these are called coiled coils. Sometimes they're called leucine zippers. Okay? So we can represent the topology of these motifs that come together in these two-dimensional diagrams. But these two-dimensional diagrams are represented three-dimensional forms, okay, that are globular in form. So often we talk about globular proteins that are proteins that have these three-dimensionally compact forms, okay, often called domains, and a protein can have one or more domains, okay? And these domains are groups of secondary structures that come together to form a three-dimensionally compact structure, okay? And a protein, like I said, can have more than one domain, okay? So we can, we'll see further on examples of proteins that are multi-domain proteins, okay? These domains are very important because there's a lot of evidence that during evolution, as well as point mutations in the amino acid sequence that creates variation in the sequence and therefore the structure and the function of a protein, you can have mixing and matching of large chunks of sequence that code for these domains. So you can have combinations during evolution as well on top of these specific mutations that changed one amino acid or a small number of amino acids. We can have transfers of large sections of a gene into another gene, and if that section of a gene that's transferred codes for a specific domain, then we can create a new protein that now has a new combination of domains. Okay, so this is a strong mechanism for evolution of protein function. over the last 4 billion years. Okay? So we have these different domains here that are shown by these different, what we call topologies. Okay? And we have names for them sometimes. So if a biochemist or a structural biologist talks about a jelly roll fold, he doesn't have to describe this complex topology here. Just the word jelly roll fold, to a structural biologist, he already knows what he's talking about. Okay? And so you can say, well, what's the difference between this and this here? Because they both have 8 beta sheets. Okay? The difference is in the connectivity between each beta sheet is different here and here. Okay? And this is just another example. Okay? Here we have a dinucleotide binding fold. It's called a Rossmann fold, which is really basically a combination of 2 motifs that have a beta alpha, beta alpha, beta, and then this is connected to another beta alpha, beta alpha, beta. Okay? And each of these units here can bind to one nucleotide. Okay? And they're often found as pairs, and these will bind to dinucleotides. Okay? So this is an example of a protein, glyceraldehyde 3-phosphate dehydrogenase, whose amino terminal domain in red here adopts this dinucleotide binding folder, this Rossmann fold. Okay? And it's binding this nicotinamide adenine dinucleotide molecule here. Okay, in this domain. Okay, and then there's a second domain in green that's shown here. Okay, so we have this sort of cartoon representation of protein structure. So this is just another protein. It can be represented as a cartoon or a ribbon form. It can show all the atoms here except for the hydrogen atoms. Okay. You can see the carbons are green, the oxygens are red, the nitrogens are blue, the hydrogens are invisible, and there's a few yellow atoms here that are sulfur atoms. Okay? It's shown in space filling form, solvent exposure form. Okay? So we have this hierarchy of protein structures. Okay? Primary structure, which is the sequence, secondary structure, which are the alpha helices and beta sheets. The tertiary structure is the topology, the way that these secondary structures come together to make this 3-dimensional globular compact form. Okay? And then these tertiary structures can come together to often make the physiologically functional form of the molecule of the protein that can have more than one protein chain in the functional form. Okay, and this is called the quaternary structure. So I just want to spend a few minutes talking about quaternary structure. So the quaternary structure describes the manner in which tertiary structures, polypeptide chains, can associate with each other to make these complexes. Okay. You can have different kinds of quaternary structures. You can have 2 or more copies of the same molecule coming together to make a polymer. Okay, and these are called Homo Polymers. Okay, it's the same molecule and more than one copy of each molecule. So Homo dimer, Homo trimer, Homo tetramer, etc. Or you can have 2 or more different proteins coming together to make heteropolymers. Okay, so it can be a heterodimer, so you can have one chain A and chain B. If they're different sequences, then we can call it a heterodimer or a heterotrimer, etc. Okay. So these hetero and especially homo polymers often show symmetry. Okay. So we often see what we call cyclic symmetry in biology. So a dimer often there will be a symmetry axis in which the rotation of one molecule, if we rotate it 180 degrees, it superimposes almost perfectly on its partner, okay? So in this case, we talk about two-fold cyclic symmetry, okay? This is an example of a protein that has this kind of symmetry. We can have three-fold cyclic symmetry, okay? So these are homotrimers, so it's the same polypeptide chain, 3 copies of the same molecule, arranging themselves in this arrangement with a threefold axis, which is perpendicular to the plane of the screen. So we can have 5-fold symmetry, 7-fold symmetry, 9-fold symmetry, 11-fold symmetry. You can have any kind of cyclic symmetry that you can imagine. Okay. There's also another kind of symmetry, dihedral symmetry, where we have one n-fold axis plus n two-fold axes that are perpendicular to that n-fold axis. Okay? So here we have a dihedral symmetry where we have a two-fold axis here, which is perpendicular to the screen. And we also have 2 more two-fold axes that are in the x and the y direction. Okay. Another dihedral symmetry is point group 3,2. So we have a 3-fold axis here, and we have 3 two-fold axes that are perpendicular to that central 3-fold axis. This is point group symmetry 4,2,2. So we have a 4-fold axis that's perpendicular to the screen, and 4 two-fold axes that are perpendicular, that are in the plane of the screen. 6,2,2. So we have 6-fold axis and 6 two-fold axes. So these are all examples of dihedral symmetry. We also have tetrahedral symmetry, octahedral symmetry, and icosahedral symmetry. Okay, that are often found in biological molecules. Okay, so this is the end of part 1. I'm not sure how much time I took. I took 9 minutes less than I was expecting. Okay, so do we have any questions about this sort of basic introduction to protein structure and nomenclature?
Yes, thank you for the lecture.
Yeah.
So in that solvent exposed nucleic structure that you show the protein, you have a solvation shell of water.
Yes.
So the water is there helping to regulate the structure of the protein.
Yes.
So this is why when you change the pH of the water or you stop on
the temperature or add co-solvents, yes.
to denaturate the proteins, this is the mechanism.
Well, because so the protein has evolved over millions or billions of years in order to get a specific sequence that is relatively stable enough for it to exercise its function in the cell in a certain time scale. So its sequence is read, it folds up quickly enough, because there's no use in evolving a sequence that after it's synthesized during translation, if it takes a couple of days for the protein to fold. So there's kinetics, thermodynamics and kinetics are both important. So in the conditions of the cell, so you have to define these conditions. 37 degrees if we're talking about a human cell, or a bacterial cell. It can be, if it's from the deep ocean, it can be very cold or very hot depending on where in the ocean, etc. So under those specific conditions, that protein has evolved to be relatively stable. It's been able to fold and exercise its function under those conditions. Now if we in the test tube produce that protein and start changing these conditions, change the pH, change the ionic strength, add denaturants, then we're going to change the balance of these forces that have been fine-tuned over millions of years of evolution to make a functional protein. So we do this kind of experiment in the lab to try and understand the basic rules that govern protein folding. So we change, like I said before, when the protein folds to make its three-dimensional structure, it's competing, those hydrogen bonds that stabilize the alpha helix and the beta sheet and everything, those are competing with hydrogen bonds with the water molecules. So you can ask, why is it that the protein stays folded? Why doesn't it just unravel and trade those hydrogen bonds, those intramolecular hydrogen bonds, with the intermolecular hydrogen bonds with water? So there's a whole bunch of arguments for that. There's one argument, or one factor, is an entropic factor. So you can say, well, entropically, which is more stable? Folding the chain or unfolding the chain? So who thinks that entropically, folding is going to be favored entropically by folding? It's favored entropically by folding. And who thinks that it's unfavored entropically by folding? Some people here. Okay, so intuitively, my first answer would be, well, it's unfavored because the protein, you've got this complex protein chain. When it folds, it only has one or a small group of conformations that are available, and that's entropically disfavorable. Okay, but upon folding, there's another effect called the hydrophobic effect. So there are hydrophobic amino acids that are usually buried. When they are buried in the interior of the protein, what happens there? Their hydration shell, the surface area of these hydrophobic residues, is now no longer exposed to the water. Okay? And hydrophobic residues normally don't like to interact with water because they're hydrophobic, they cannot make hydrogen bonds with water. So therefore, a water molecule that comes up and gets close to a hydrophobic region of a molecule that cannot make hydrogen bonds with itself, now this water molecule has to rotate and make a hydrogen bond with the hydrogen molecule next to it. It's not, its entropy is reduced, okay? Because in both water, if a water molecule has many water molecules around it, in any direction it can make a hydrogen bond, okay? But if it comes up to the surface of a hydrophobic protein, now it can't make this hydrogen bond. So it has, now its entropy, its conformations, the number of conformational states available to that water molecule is reduced. So one sort of general idea about protein folding is that the first step is sort of like this collapse of the protein where all of the hydrophobic residues get buried in the interior. So the three-dimensional structure will generally favor a configuration where the hydrophobic residues are in the inside and the hydrophilic residues, those amino acids that can make hydrogen bonds with water, are on the surface. Okay? And that liberation, so the burying of hydrophobic surface liberates water molecules. Now they don't have to interact with that hydrophobic surface. They can interact with other water molecules in the bulk solvent. So that entropic effect compensates the reduced entropy of the actual protein molecule. Okay? So there's this very, very delicate balance between if you add up all of the interactions all of the favorable interactions of an unfolded protein with the solvent. That's a big number. And if you add up all of the favorable interactions of a protein with itself when it's folded up, plus those interactions on its surface with the solvent, that's also a very big number. And the difference between these two big numbers is that difference is the energy that is stabilizing this three-dimensional structure. And that is a very, very small number of, okay? So that poses the big challenge for physicists like you to try and calculate under physiological conditions what the behavior of a protein is going to be. Because you're making your decision based on very, very small differences between big numbers. And therefore, any small error in the calculation in the summing up of all of these small, weak interactions, okay, can change the balance, okay? And that's the big challenge that we have when we try and first calculate the structure of a protein from a sequence, and then that's just the first difficulty, because we get a structure, but really we know that for a protein to function in the cell, it's not static, it's dynamic. So in fact, what we have to do is calculate a family of possible structures and see how the interchange between these conformations is coupled to how that leads to it manifesting its function in the cell. Okay, so a protein acts as a catalyst, as an enzyme, because during the catalysis of the enzyme of the reaction, it binds to the substrate, and then it modifies its conformation and the substrate's conformation during the transition state of the chemical reaction. Okay, so this conformational flexibility of the protein is very, very important. Okay, so that's also a challenge. So we can get structure of a protein from crystallography or another method, and then we have to simulate how it can evolve from that static structure that we determined experimentally, and what are the other available structures that are theoretically possible, and how can we rationalize that in terms of the function of the protein. Any other questions?
Maybe shed some light into this asymmetry that there seems to appear due to entities. There's a different.
Oh, yeah.
I don't see an explanation here.
Well, if you look at the Ramacandra plot, well, if we look at amino acids first, amino acids, structure of an amino acid. Okay, so let's look at the most simple amino acid here. Well, not the most simple, but it's the second most simple amino acid, alanine here. So it has this chiral center here, this alpha carbon. Okay, it's shown that these three atoms are in a plane, but they're not in a plane. It's a tetrahedral orientation around this carbon. So there's a hydrogen atom that's being shown that's not shown here. Okay? And these are L amino acids. So all of the amino acids are L amino acids. So if you remember your chemistry, there's a mirror image of this. It's a D amino acid, and basically the hydrogen atom, instead of being behind the plane, it's going to be above the plane. Okay? So each amino acid has a certain handedness. So all proteins are made up of L amino acids. So when we consider the availability, so here we have here are the hydrogen atoms. They're shown here actually. Okay, these small spheres here. Okay, it's behind the plane. Here it's above the plane, behind the plane, above the plane. Okay. So in the Ramacandra plot, we have this right-handed alpha helix here, which is and this is basically, this here is the same conformation, okay? with the psi and phi angles inverted, okay? So this will be a right-handed alpha helix, okay? So if we look at the main chain, of the alpha helix, this is going to be sort of like the mirror image of this, okay? Well, it's not going to be the mirror image. This is a left-handed alpha helix made by L amino acids, okay? Now, if we make a protein made up of D amino acids, so we can synthesize this in the laboratory, and it's been done before, okay? And what you will find, the Ramacandra plot for a protein made up of D amino acids is this image inverted. So we have this large region here, this small region here, and this beta sheet region here, okay? And all of those proteins have left-handed alpha helices, okay? And they fold up actually just as mirror images of their right-handed proteins. And if they're enzymes, they will catalyze the reactions of substrates that are their mirror images, okay? So this asymmetry here in this Ramacandra plot is a consequence of the asymmetry of the constituent amino acids. Okay? So yeah, there's another question. Yes? There's two questions. Okay. Yes?
I'm learning English. Do you know alpha sheet?
Alpha sheet? Beta sheet? Beta sheet or alpha sheet?
Alpha sheet.
No, I don't know what an alpha sheet is. I know what a beta sheet is. What's an alpha sheet?
New structure.
Yeah, I've never heard of it, but I might be out of touch. What does it look like?
Structure amyloid.
Ah, okay. Amyloid structures. Okay. Amyloid structures are
Alpha sheet.
they're alpha sheet. They're called alpha sheets. Well, I always thought that most amyloid structures are beta sheets. Well, they look like beta sheets, but they're often so they can be protein that normally has many alpha helices can unfold and then adopt a new structure that is dominated by beta strands. That's yeah. So okay. So there's a lot of interest in understanding pathologies that are caused by misfolded proteins. So proteins have also evolved so that they don't misfold. Okay? So you want the protein to be synthesizing itself, and you want it has to fold into a form that's going to be functional. And it can't, if it also folds into a form that causes problems for other processes in the cell, that is going to be evolutionarily detrimental. The cell is going to be sick, okay? And therefore, that cells that have a gene that codes for a protein with that kind of dysfunction will usually be eliminated. So there's a whole bunch of like these evolutionary filters over millions and billions of years that go into defining what a protein sequence is being used by the cell. So it has to be able to fold quickly, it has to be able to execute its function, it can't misfold, it can't interfere with other kinds of reactions. There's a whole bunch of different functional filters that evolutionarily, will be eliminated. These dysfunctional forms will be eliminated. Okay, so there are many diseases that affect people, old people like me, in their old age, because we often start having problems with proteins misfolding in important parts of our body. And there are we call it sort of like many different processes in the cell that that seem to be able to detect misfolded proteins and eliminate them, tag them, and then send them to be degraded. Okay? But as we grow older, these processes that are able to correct these errors in metabolism are often less and less efficient, okay? And it basically escapes our control, okay? This happens both at the protein level and at the genetic sequence level. So we also accumulate mutations. We have systems that detect mutations in the DNA sequence and correct them as well. But these errors accumulate during our lifetime, and it gets to a point that sometimes the errors occur in certain genes that affect our ability to correct these errors. So if we get mutations in the processes that correct errors in genetic replication, then we start accumulating more and more mutations, and then we have problems as well. Okay? So I'm not sure if I really answered your question. There is one more question.
Usually when we want to simulate protein, we differ this from crystallography or RNA experiment. But this is hard. And can you tell things about the theoretical and computational efforts to predict proteins?
Oh, well, I can't. I think there are going to be other lecturers in this course that are going to talk about protein simulation of protein function. Oh, sorry, protein dynamics, etc. Okay, I'm not an expert in this. I'm not an expert. Well, I mean, there's just so many. You can have a whole career in this. Basically, what you will do is you will try and set up an algorithm that will calculate all of the forces between pairs of atoms. It depends on how fine-grained you want to make your simulations. If you want to simulate something in high detail, you have to be able to simulate interactions between all. Well, you can say I want to simulate the interactions between every electron in my structure. Okay. In that case, you have to use like quantum mechanics. Or you can say, well, let's assume that you can simulate the behavior of each atom, so you can simulate the interactions between pairs of atoms. Or you can say, well, let's treat each amino acid as a sphere, okay, with a different radius based on the size of the atom, and maybe give it another kind of characteristic, if it's a hydrophilic or hydrophobic, or if it's positively charged or negatively charged, you can give it a certain character. So you can make these fine-grained or coarse-grained simulations. Obviously, you're going to gain in speed, but you're going to lose in the fine details, and sometimes these details are very important. Has everyone heard about the Leventhal paradox? Yes. So there are literally more possible conformations for a protein, or even a small protein, than there are a number of atoms in the universe, I think. Okay? But things like the Ramacandra Plot allow you to eliminate a lot of these possible conformations. And then other things like the kinetics of protein folding, or things like the hydrophobic effect, will allow you to eliminate other types of conformations. Okay? But I think you're going to get delve deep into simulation of protein behavior, structure, and dynamics in the talks of the other lecturers in this series. Any more questions before I go to the next? Okay, so basically, following up from your question about protein structure, My next lecture is going to be about what are the main techniques that we use to determine the structures of proteins. Okay. So, protein structure determination. So, I want to take the next period to talk about these methods of protein structure determination. I'm going to concentrate on these first three methods here, which allow us to obtain models at a relatively high resolution of the structures of macromolecules. Okay, and these methods are X-ray crystallography, nuclear magnetic resonance, and cryo-electron microscopy. So I'm just going to basically give a small introduction about more or less the kind of data that you get from these experiments and how they're related to the structure. And obviously, to understand in detail how these methods actually work, you have to take several courses and study for several years to understand these at a high level of detail. But I'm just going to sort of like, I hope I'll be able to pass to you the nature of the data that you collect, and therefore how that, what kind of structural information are you really getting from these kind of models? Okay. So, people that are involved in determining the structure of these macromolecules, they participate in an area of biology called structural biology, and this has been very, very important. The X-ray crystallography, NMR, and cryo-electron microscopy allow us to obtain models that have resolutions below 1 nanometer. So, sub-nanometer resolution allows us to see the positions, to resolve the positions of atoms, or at least of molecular groups, of functional groups in a molecule, okay? Remember that carbon-carbon bonds are separated by, the carbon atoms and the carbon-carbon bond is separated by 1.4 angstrom resolution. The diameter of an alpha helix is around 10 to 15 angstroms, okay? Which is 1 or 1.5 nanometers, okay? And you can start seeing alpha helices and beta sheets at resolutions below 5 or 4 angstrom resolution, okay? So, there's a lot of importance, or a lot of credit given to people that have worked in this area of structural biology. Okay, a lot of Nobel Prizes that have been given out by people who have developed and used these methods in order to obtain information of macromolecules. So, we have the Nobel Prizes to Röntgen and Lowy, and the two Braggs for discovering and interpreting the interaction of X-rays with matter, especially with crystals. So the Braggs were able to interpret the X-ray diffraction patterns and use this to determine the positions of atoms in crystals of small molecules, salts, etc. And then other scientists over the decades, the first half of the 20th century, tried to interpret the diffraction patterns of protein crystals and DNA crystals, okay? Pauling, in fact, he was interpreting he was a crystallographer determining the structures of small peptides and small molecules, but he also had a very good appreciation of the geometric constraints of a polypeptide chain, and so just from theoretical considerations, proposed the structures of alpha helices and beta sheets even before their structures were determined experimentally. The first protein crystallographers that were able to determine the structure were Perutz and Kendrew that determined the structures of hemoglobin and myoglobin. And in the same laboratory in Cambridge, Watson and Crick, using data from Wilkins's laboratory, were able to propose a model for the structure of the double helix of DNA, okay, based on X-ray diffraction pattern of DNA fibers. They weren't crystals, they weren't perfect crystals, they were DNA fibers. Dorothy Hodgkin determined the structure of insulin and several other important biological molecules in the 60s. These three German researchers were able to determine the first membrane protein, a photosynthetic complex. Kurt Wüthrich was a pioneer in the use of NMR for determining the structures of proteins. We'll get to him later on. McKinnon and Agri, they were the first, they determined the structure of a membrane channel, so a membrane protein that produces a conduit for ionic, in their case, ionic substrates to cross the hydrophobic barrier of a cell membrane. So this is very, very important in biology. Roger Kornberg determined the structure of RNA polymerase. Got this protein, the Nobel Prize. So this molecule is very, very important in the process that converts the DNA sequence into RNA. He's the son of Arthur Kornberg, who discovered the function of DNA polymerase in the 1950s or the 1960s. Okay? Venkatraman, Ramakrishnan, and Stites and Yonath, they got a Nobel Prize for determining the crystal structure of the ribosome, which is a huge complex of dozens of proteins and several RNAs that mediate the synthesis of proteins in the cell. Okay, so these are all central, important processes in biology. We understand how they work at the molecular level because of the work of these structural biologists. Okay? Lefkowitz and Kobilka got the Nobel Prize for determining the function and the structural relationships in these G protein coupled receptors that are targets for many drugs in modern medicine. Okay? Talking about simulation, Karplus, Levitt, and Morschel got a Nobel Prize in 2013 for developing multi-scale models for complex chemical systems. This is just a fancy way of talking about developing algorithms to model protein behavior or polymer behavior, okay, not just protein, but the behavior of polymers in computers. Okay? And just recently, a couple of years ago, 3 years ago, Dubochet, Frank, and Henderson got the Nobel Prize for their contributions to developing certain methods that have allowed cryo-electron microscopy to obtain high-resolution structures of macromolecules. Okay? So there's been a lot of recognition over the years, over the last century, to the contributions of structural biologists in important areas of chemistry and in biology, in medicine, physiology and medicine. Okay? So crystallography, for example, these structural biology methods allow us to determine structures of very, very complex proteins. Okay? So this is the respiratory complex #1. So you biologists in the crowd might be able to recognize this from the metabolism, the electron transport chain that occurs in the mitochondria. So this is we have a mitochondrial membrane here, several different proteins in different colors that are found embedded in this membrane. And we have this large globular domain here, or globular section here, that is made up of several different subunits. And so we're able to crystallize these complex particles that we can isolate from cells. So we're able to develop methods to purify these very, very complex forms to a degree that we can actually crystallize them, or we can study them by other methods. Okay? So, protein crystallography, basically, you have to be able to crystallize your protein. That's the first obstacle that you find. And these crystals can come in different forms. Okay, they can be long filaments. You can have nice plates. Okay. But basically, what is a crystal? So in X-ray crystallography, you have the molecules arranged in a three-dimensional lattice in which you have a repetition of one or more molecules. Their arrangement among themselves is repeated many times in the lattice. So you have what we call a unit cell, okay, the contents of which can have one or more molecules. And a crystal is basically considered a repetition of a translational repetition in 3 dimensions of this unit cell, okay? And the fact is that in protein crystallography, the protein crystals are different from salt or sugar crystals. If you get salt or sugar crystal and you dry and you put it on the table and you try and crush it, there's a lot of resistance. It's very hard to crush, okay? Protein crystals, they are held together by very, very weak interactions, non-covalent interactions between large molecules, and there's a lot of space between them. There's a lot of solvent, okay? You can have like sometimes over 70% solvent. So they're very, very soft and very, very fragile. You can cut them sort of like butter. You can get a whisker, a cat's whisker, and touch a crystal and you just break apart, okay? And there's also very much interest in determining the structure of membrane proteins, because many of the most important biological processes occur in proteins that are associated with or embedded in the membrane. And this presents a very, very big challenge to a biochemist. To first purify a protein from a membrane fraction is very, very difficult because it will often have a part of the protein that is exposed to the solvent, exposed to water, but it will also have another part of the protein that is most accustomed, that folds only properly in the hydrophobic environment of a membrane, of a lipid membrane. So you have to purify it and maintain these interactions between the lipids or detergent molecules that are replacing the lipids during the purification process. You have to purify it so that the conformation of this region is also maintained. So this is very, very difficult from a biochemical point of view. And then trying to get this kind of molecule to crystallize is also very, very difficult. So this is a very, very big challenge. So like more than 90% of the protein structures are the structures of proteins that are soluble proteins, that are not associated with membranes. Only a very small fraction of the structures of proteins that we have in the databases are membrane proteins, okay? So how do we actually try and crystallize a protein? Well, we mix the protein, we purify the protein, usually, and we try and get it as concentrated as possible without it precipitating, okay? And then we mix it. So here we have the protein in this green solution. Here this is our protein. And then we mix it with a solution, another solution that has some kind of co-solvents that reduce the solubility of the protein. So we have a small drop of a couple of microliters or a fraction of a microliter of a protein, and then we mix it with a drop of a solution that has something that reduces the solubility of the protein. So in the first moment of mixing these two drops, the first thing that happens is that the protein is diluted by 50%. So it would say 10 milligrams per ml. When we mix it with an equal volume of this solution here, now it's 5 milligrams per ml. So it's no longer, now it's less concentrated, okay? But it's in the presence of something that reduces its solubility. So there's several things that can happen here. It can immediately precipitate, or it can not precipitate. So, and then what we do is we immediately put this, we flip this slide. We can do this in a number of ways, but in this diagram, we flip this glass slide over, and we put it inside of chambers. This drop now is inside this sealed chamber, in which this drop is in equilibrium with this solution here, okay? And this drop here has 50% of the concentration of these co-solvents. And so now, what we're going to have is we're going to have a process that's called vapor diffusion, okay? Due to the higher vapor pressure of water in this drop here, because the concentration of the co-solvents is less here than here, we're going to have a net migration of water, of the volatile components of this drop, which is usually water, okay? They're going to migrate from this drop to here until the concentration of co-solvent here is equal to this. So at the time equals 0, this concentration of the co-solvents here is 50% of this. But over time, the volume of this drop is going to get reduced over a couple of days, a couple of weeks, okay, depending on the conditions of the experiment, the temperature, etc. So over time, this drop is going to get smaller and smaller until it gets to around the original volume of this drop here. So until it gets to be 50% of the volume of its original volume. Okay? And during this time, the concentration of the protein is going to go up. The concentration of the co-solvents is going to go up. And maybe during that time, the protein is going to become supersaturated. And it will start to crystallize. That's under the best conditions. That's the idea behind it. Okay? So this is like a phase diagram of this kind of experiment where we have at time 0, when we mix the two together, we're in this region of concentration, of protein concentration here. So this is the original protein concentration here, and this is the original precipitant concentration here. We mix them together, the protein concentration goes down by half, the precipitant concentration goes down by half, so we're in this region. We're in a region where the protein is still soluble, but over time, during this vapor diffusion, the volume is going to go up, so the protein concentration goes up, the precipitant concentration goes up, so we start going in this direction here, and we go into a region where the protein becomes supersaturated, and it will start to crystallize. Now, if y'all remember your classes about crystallization, there's two different processes in crystallization. There's nucleation, and then there's growth. And nucleation is a much, it's entropically less favored, and it is, so it's kinetically slower than the growth. So once you get a nucleus and you just have to add more molecules to the surface of the crystal, that's faster. So sometimes this process of vapor diffusion, before we start crystallizing, sometimes it goes deep into this region of supersaturation before the crystals actually start growing, just due to the slowness of the nucleation process. And then we start getting a crystal forming, and then, oops, and then when the crystal, so then when we get the crystal forming, then the protein concentration is going to start going down because the protein is starting to get, it's going into the solid state of the crystal. And it will keep on growing until the protein concentration is less than the solubility, okay? And then we'll get, if we're lucky, we'll have these crystals. So if we have these crystals, we'll be very happy, like Christianity is happy here, and then we'll take the crystals to a synchrotron, okay? This is the Brazilian synchrotron. This is the actual Brazilian synchrotron. There's a new Brazilian synchrotron being built in Campinas. But you take it to a synchrotron facility and you expose the crystal to X-rays, and the X-rays will interact with the crystal and form a diffraction pattern. Now, the diffraction pattern is a result of the interaction of the X-rays with the electrons in the crystal. But because the crystal is repeating in 3 dimensions, there are certain angles. There are certain angles in which the diffracted rays will interfere constructively and create spots, okay? We call them reflections because very simplistic manner of interpreting the diffraction pattern is that the X-rays are being reflected by imaginary planes that connect the lattice points in the 3-dimensional crystal. So you can imagine a 3-dimensional crystal is made up of these equivalent points, these equivalent lattice points, okay, that are the corners of the unit cells in the crystal, okay? And you can imagine imaginary planes that go through these points, okay? And you can cut this lattice in hundreds of different manners to make different families of planes that go through the points. So here we're showing planes that are going through the points. They're parallel planes. Here they're going through points going in a horizontal manner. But you can also imagine parallel planes going in a vertical manner. You can imagine parallel planes cutting across here at a 45-degree angle. You can imagine parallel planes going through this lattice point and this lattice point, and this one and this one and this one and this one. So you can have many different types of planes that will reflect these X-rays, and for each set of planes, that there will be a certain angle in which the reflected rays will interfere constructively, and that will generate this diffraction. Now, obviously, these planes are not from these imaginary lattice points, but in fact, what they are, it's the sum of the interactions with all of the electrons in all of the atoms within the unit cell. Okay. But since these X-rays are interfering with each other in a predictable way, okay, you can imagine that the sum of for each unit cell, the sum of all of the diffracted X-rays caused by each electron in the unit cell, they will sum up to make a resultant diffracted ray, okay? And so we can treat the resultant diffracted ray as one unit, or one wave, okay? So from this diffraction pattern, there's a mathematical relationship between the electron density, the distribution of electrons in the unit cell, and this diffraction pattern. And this is the form of this mathematical relationship. It takes the form of takes this form, where the electron density is the sum of this is the structure factor of basically the amplitude of each of the reflections, and an exponential function that has a x, y, and z coordinate in the electron density, and an HKL, which is the coordinate of each of these reflections. So this takes the form of a Fourier transform. Okay, so the diffraction pattern is a Fourier transform of the unit cell. Okay, so if the electron densities, this is the electron density, these clouds here, this electron density, this diffraction pattern, okay, which is represented by this structure factor here, which is basically the intensity of each of these points here, okay, is a function of, is the Fourier transform of the electron density. Then the electron density is the reverse Fourier transform of the diffraction pattern. So if we have the diffraction pattern, we can apply this equation and calculate this electron density map. Now, in order to apply this equation, what we need is this structure factor here. Now, this structure factor is actually a vector. And it's basically a vector, and the direction of the vector is defined by this HKL. This HKL is basically defining the angle at which it's defining the set of planes that are cutting this crystal lattice. Okay? Like I said, there are a whole bunch of different ways you can cut the crystal lattice. Each form is defined by an index HKL. Okay? Now it also has, so it has a direction and it also has a phase. Now the phase here, unfortunately, when we do the experiment, the phase is lost in the experiment. So we have the intensities, we have the amplitudes of each of these waves, okay? And we have the frequency, which is determined by the HKL index, but we don't have the phase, okay? And in order to construct this electron density map, okay, it's basically the sum of many, many different waves. Each wave is defined by each of these spots here, okay, with frequencies in each of the 3 dimensions given by HKL, okay, amplitudes in each of the dimensions given by the intensity of these spots, but plus a phase. In the phase we've lost, we don't obtain this in the experiment. So we have to be able to calculate the phases in a different way. There are several different ways that we calculate the phases. We don't have time to get into that. But if we are able to determine the phases, now instead of having a black and white picture here of the electron, of the diffraction pattern, we get sort of a colored representation of the diffraction pattern, where each spot now, as well as having an intensity, has a color, which is basically its phase. It's the phase of the wave. Okay? And now what we can do is basically add up all of these waves. Okay? The waves that are close to the center of the image have very low frequencies. Okay? And they give us information about the general repeating high density, low density oscillations in the crystal, okay? And we have waves that come from the reflections at the perimeter of the diffraction pattern that are high frequency waves that give us high detail information about the details of the structure, okay? So basically what we do is we add up literally hundreds or thousands of these waves, okay? Because this is one image. What we do is actually we rotate the crystal during the experiment, and every 1 degree or every half degree, we collect another image. So this is actually, this diffraction pattern, it's not 2-dimensional, it's actually 3-dimensional. So we collect hundreds of images, okay, and that allows us to get information for thousands or tens of thousands, or sometimes hundreds of thousands of reflections at a high redundancy, so we can calculate their intensities with a high accuracy, okay? And then using other methods, we calculate the phases, and that allows us to do this summation here. And you can see that we're adding up a whole bunch of just simple sine or cosine waves with different frequencies, and that is creating the summation is creating a map. Here it's a 2-dimensional map, it's a 3-dimensional map because it's 3-dimensional waves, okay? And this gives us a 3-dimensional electron density map. Now, so if we can collect a lot of these reflections, including reflections with high indices that give us a lot of information for high resolution information, we can, the resulting map can often be very, very detailed, okay? And that will allow us, so that's what we get. We get the map. And from the map, we have to be able to construct a model. The map is going to guide us to make a chemical model that fits the map. The chemical model, we use information that we've learned from our general chemistry knowledge. We know that carbon-carbon bonds in an aromatic, in an indole ring have a distance of 1.4 angstroms plus or minus 0.01 angstroms or something like that. So we can make restrictions on the distances between atoms in our model. We can use the map to restrict the possible positions of these atoms. We can use our chemical knowledge and the sequence of the protein to make a covalent structure that we're going to fit into the map, okay? So what we're going to do is we're going to try and balance the quality of the fit of our model to our map, and with the quality of our model vis-a-vis our chemical knowledge of the behavior of our knowledge of what we can reasonably tolerate for all the different kinds of bonds in a protein molecule or in a DNA molecule or whatever we're trying to model. Okay, so we know from basic chemistry what the geometry of a aromatic side chain is. All of these atoms have to be in the same plane. Okay, these angles have to be in a certain range, etc. Okay, so we can use that. We can try and balance the fit and the quality of our model, the chemical quality of our model, with the fit of the model to the electron density. Okay, so that's basically what protein crystallography is going to allow us to do. Now, you can see that this is a very, very high resolution electron density map. We can see the carbon atoms. We can't see the hydrogen atoms, but we can see sort of like these little bulges here where it seems like there might be like a hydrogen atom attached. And we can see these spheres here, which are water molecules. So on the surface of a protein inside a crystal, often they're like 50% water. on the surface of the protein, there are water molecules that are interacting strongly with the groups, the functional groups on the surface of the protein. And these interactions between water and protein groups will reduce the motion of these water molecules. And the water molecules, they will be strongly bound to these groups by way of usually hydrogen bonds. And in high resolution protein structures determined by crystallography, you can often see one or more, okay, water molecules bound per amino acid. So a protein that has several hundred amino acids, if it's a high resolution crystal structure, there will also be several hundred water molecules included in the PDB file that will show the positions of these water molecules. Now, the water molecule is usually, the density, what we see usually is a sphere, even though we know that water molecules are not perfectly spherical. The resolution doesn't allow us to see the asymmetry in the water molecule, and so it's usually just represented as an oxygen atom, even though we know that there are two hydrogen atoms attached to it. Okay? So here we'll see the structure of a protein, and these little X's here are water molecules that are strongly bound to the surface of the protein by way of hydrogen bonds. Okay? Now, so that's crystallography. Any question about crystallography?
I have a question. How do you determine the resolution of PDB models? How is the...
Yeah, well, the resolution is determined by at what point do you can sort of like tell generally that the intensity of the dots in the center of the image here are more intense than the ones on the edge. And you can see out here, we just all of a sudden lost, we don't see any more dots here. Okay, so these dots here are going to determine the resolution. They have high indices, okay, they're HKL indices of this dot, they're big numbers, and that means that we are slicing up the unit cell in many different slices, okay? Dozens maybe. We have parallel planes that are very, very close together, and that gives us a high frequency, a high resolution information about the protein, okay? So that's how the resolution is determined. Yeah?
So, in terms of using usual external diffusion versus the synchrotron radiation, what is the, like, you get better resolution? It's what, the intensity is higher? Or
Yeah. So you can have a home X-ray source. Like in my institute, we also have an X-ray diffractometer, okay? But the intensity is very low. It's much lower, and the size of the beam, the cross section of the beam is larger. So what you do, if you have a low intensity beam and you have a beam that's larger than the actual crystal, okay? So it takes more time to collect the data. Okay, so you have to expose the crystal to the X-rays for several minutes, and then rotate it 1 degree. Actually, during that several minutes, you're rotating it for 1 degree, and then for another several minutes, you rotate it for another degree. So that might take you a couple of days to collect the data set.
Total damage?
Well, it's not going to damage that much because the X-rays are low intensity, and it's frozen. So you maintain it frozen, so they're not damaged. But the quality of the beam is, if you're exposing the whole crystal, the crystal is never perfect. So really the crystal, any crystal that you can actually mount and see, it's actually not one crystal. It's got several different regions of the crystal that are sort of a little offset with each other. And these imperfections destroy the quality, reduce the quality of the data. So in fact, here, all of a sudden, you have good data, and then all of a sudden it gets bad. Okay? That's probably because there's some kind of imperfections in the crystal that beyond that point, you just get destructive interference between different domains of the crystal. Now, in the modern synchrotrons, the X-ray beams they're like micro beams, okay? And so you can illuminate just a small part of the crystal. And therefore, you can like just illuminate just like the tip of the, like if you isolate one of the leaves of here, you can illuminate just a small tip of it. And that region probably has a higher chance of being perfect. And so the quality of the data is going to be higher. So in our institute, to collect a data set, you have to take several hours or maybe a couple of days to collect one data set. At a modern synchrotron, it takes like a minute. So it goes really fast. You don't even have time to relax. At least when you use in my lab, you can start the data set and come back the next day, and or here you can't even have a cup of coffee. So it's high intensity work. Okay. Yeah.
When you have the crystal, the X-ray have a reflection, what happens in terms of the interference that you can have when the X-ray is going out? It's producing a reflection, and you have more electrons. I don't know if I am not physicist, my question is, if you have this reflection, okay, you have an electron, you have an interaction, and then it's going the X-ray. So it means that you will have another electron in the reflection. How is the tracking of this interference? Or have some interference? I don't know.
There is a good explanation for why is it that it's escaping me now. Why is it that the intensity of the reflections at high angles is less? I can't remember exactly why. But essentially, what you have to imagine is that I don't want to get into the theory. The physicists here will correct me with all the things, but basically, if you have an X-ray going through the crystal, it's an oscillating electromagnetic field, and that oscillation in the field will cause the electrons to oscillate, and therefore and when the electrons oscillate, each electron becomes a source of X-rays. So the oscillating electrons, you can think of each electron individually, or you can think of the sum of the electrons in the atom, or etc., or the sum of the atoms in the unit cell, but they will all oscillate and emit in all directions. And so you'll have different waves, all from different sources, the source being each electron, and they will be interfering in different directions, so depending on the angle of the incident beam the beam leaving, okay, the reflected beam, you're going to have certain directions in which you're going to have constructive interference. Only certain specific directions will be constructive. All the other directions are destructive. That's why you have all these blank, 90% of the surface of this pattern here has no intensity at all. It's just background. Theoretically, these should be very, very intense, very, very small points. But because, like I said, the crystal is not perfect, so these reflections are not perfect points. They're sort of like real fuzzy, okay? Now there is a good physical explanation as to why the reflections at the very edge have lower intensity, but I can't remember exactly what it is. But there is a good physical explanation for Other questions? Yes. Yes. Oh, yeah. Well, nucleation. Well, nucleation can take, depends on the crystal. It can be very fast. So we've had examples where it crystallizes as soon as you do it. You mix them, look at it under the microscope, and you see the crystals. And then there are other experiments where it takes several weeks to nucleate, to make crystals. There's a problem. Sometimes instead of crystallizing, it just precipitates. Okay, so yeah. Oh, yeah. So we don't know. If you knew, you could make a lot of money. We basically have to test literally hundreds of different conditions, just on a trial and error basis. We have to test. We have the plates of 24, 96, 384 well plates, where we mix protein and precipitant, different cocktails of precipitant recipes. You can buy them. You can buy kits that have, 48 or 96 or more different cocktails, and you can test them. And if you're lucky, you'll see a couple of them that might give you some crystals. They might not be very nice crystals, but it might be a crystal that seems reasonable. And then what you can do is you can get that condition and start tweaking with the conditions. You can increase or decrease the concentration of the components in the mixture and try and make the condition. You can try and get it so that it can crystallize more efficiently. So if I find that I get I do an experiment with, 20% polyethylene glycol and 500 millimolar ammonium sulfate, something like that. Okay, these are two things that will cause precipitation. And pH 7. Okay, and I find that I get these really crummy crystals that aren't very nice. Okay, or it gets, that's the best thing. That's my best result. What I can do is I can do an array. I can test a whole bunch of different experiments. 24 different experiments where I can change the pH, go from 8.5 to 6.0. I can change the polyethylene glycol from 23% to 17%. Okay? I can start changing these things, and then hopefully maybe one of these conditions, now all of a sudden I get nice big crystals. Okay? So first you have to try and identify some kind of condition that gives you some kind of reasonable crystals, and then you start tweaking with that, that condition to try and optimize the crystallization conditions. Okay? Okay, so, any more questions about crystallization? Yeah.
How do you get the scheme map with the Fourier transformation? It's an iterative
Yes. Yeah, it's iterative because, like I said, we have to calculate the phases. And the quality of the map depends on the quality of the phases. So if you don't have, if the phases aren't correct, the map is just garbage. And we can't build the model. So like I said, there's a whole, it'll take you a whole course to learn about how you actually calculate the phases. There are different ways of doing that. But initially, the phases usually they're not very good. And often the phases are only good for these low frequency reflections. The higher intensity ones, but the quality of the phases for these reflections are usually not very good. So that allows us to make a map that's a very low resolution map. So therefore, my map is not this. It's not such high resolution. My first map. But if it's good enough that I can actually see regions where there is protein and where there is no protein, or I can say, this is an alpha helix, this is a beta sheet. So I can make a rudimentary model that maybe doesn't even have the side chains, but it just has the main chain. I can say, and that model will allow me to calculate phases now based on a more detailed model. Okay, so now I can use this relationship. Like I said, the electron density is the Fourier transform of the diffraction pattern, but also the diffraction pattern is the reverse Fourier transform of the density. And the density, if I have a model, I can calculate the density. So if I have a rudimentary model here, I can calculate a rudimentary density and calculate what the diffraction pattern should be, including the phases. And then I can use those phases with my experimentally determined intensities here to recalculate the electron density and make a better map. And then that can be used to make a better model. And then that better model can be used to recalculate the phases. Okay? So I can go back and forth until I get to the point where calculating the phase, and I'm always comparing the intensities that I observe experimentally with the intensities that I calculate theoretically based on my model. And that will tell me if my model is actually getting better and better. Okay, and then I'll get to the point where I can't adjust the model anymore. That I can't adjust the model. I can't improve the correspondence between the observed intensities and the calculated intensities. Okay, but the phases initially are determined in some kind of experimental way, indirect way, and then once I get my model, then I make the phases are based on my model. Okay, and then I adjust the model in order for using those phases. I adjust the model in a way that makes the intensities match the experimental intensities. The calculated intensities match the experimental intensities. So I keep on going back and forth in what they call real space here and reciprocal space here, because there's a reciprocal relationship between the electron density and the Fourier transform. That's and the diffraction pattern. Any more questions? Yes.
I want to know the crystallization process if you want to change the structure of
Say that again.
during the process you change the structure
Yes. Yeah. Okay. The question is, during crystallization, whether the structure of the protein is affected. Yes, definitely. Because the proteins, when they crystallize, the protein molecules are interacting with other protein molecules. Each protein. It's like you're sitting here in this crowded region of the audience, and each of you is in contact with the protein with protein molecules around you in three-dimensional space. These contacts can and will affect some aspect of the structure, because the protein is first crystallized, so what you're doing is you're basically capturing the protein in one arrangement that is conducive to crystallization. And then what you're doing is you're freezing it. So actually, when it's frozen, it's no longer moving around very much. And so you're determining your data based on this sort of frozen configuration that is favorable to crystallization. So during the crystallization process, like I said, there's protein, it's in equilibrium with, this is nucleation. And then we have growth. So we have lots of protein. Okay? We have protein protein molecules being attached to the surface. Okay? So this process, this growth, this nucleation of the growth, if this protein is actually, it can exist in several different conformations. It can exist, say, I don't know, let's say it's got 2 domains. It can exist like this, or it can exist like this. Okay, so it's an equilibrium between these two. But maybe this form nucleates and this form does not nucleate. So what I'm going to determine is this structure here. I'm not going to determine this structure here. But this structure might actually be physiologically relevant. And this might just be sort of like a dead-end configuration that's being trapped during the crystallization. So you have to be careful about interpreting the data from a crystal structure, especially if you're making conclusions about relationships between regions that might not have very strong interactions. So if I see that in my structure, the interaction between this region and this region here is very weak, and that this region is in fact in the crystal, it's in contact with another molecule, okay, this region here is in contact with this, maybe I should think, maybe this relationship between this part of the protein and this part of the protein, that's only connected by this sort of feeble little linker here. This might be due to the interactions between this region and this in the crystal. Okay? And then maybe in my molecular dynamics simulation, I actually find that these contacts here are actually falling apart. And that might actually be physiologically relevant. Okay? Now, you can get experimental information. You can get experimental evidence for this kind of thing. And that's in the next topic. Okay? So crystallography will give you high resolution data for a structure in one configuration, usually in one configuration. Now, if you're very, very patient and you try and crystallize, you can try and crystallize the protein in different forms, or with different substrates bound, or with inhibitors bound, etc. And you can try and get different structures for the protein in different states. And this will allow you to have an idea of what possible conformations are available to a protein. Yeah. Oh, we got a break for lunch. Okay, so I went slow and I went fast in the first half, and I went slow in the second half. Okay, so maybe I'll talk about this at the end. Okay, yep. Okay.